series, we will speak with scientists from the Dangoor Center for Personalized Medicine to gain an up-close understanding of what happens in their research laboratories, which developments are expected to reach the market, and how medicine will look in the coming years. This time, we're hosting Professor Yossi Mandel. With us today, Professor Yossi Mandel joins us from the School of Optometry and Vision Scientists in the Faculty of Life Sciences, where he is the head of the school, and it's associated with the Dangoor Center for Personalized Medicine. Professor Mandel's lab focuses on developing miniature devices implanted in the retina that restore vision for individuals who have lost their sight due to retinal diseases. He's an ophthalmologist with a doctorate in bioengineering from the Hebrew University of Jerusalem. He completed his postdoctoral fellowship at Stanford University from California and joined Bar-Ilan University in 2013. Currently, he's involved in technological advancements to improve the vision of individuals affected by blindness-causing diseases. One of his studies recently received a prestigious research grant from the European Union. Professor Mandel, thank you for joining us today.
Thank you. Thank you for hosting me. Yeah.
We appreciate having you here. Perhaps we'll start with a little bit of background for people who are not as familiar. What exactly is the retina, and what role does it play in vision?
So retina is actually the sensing part of the human eye. So it is in the back of the eye, and it serves as a kind of a combination of a camera and a very good image processing. Sometimes people refer to the eye as a camera, and I always say this is like, downgrading the retina in the eye, because it is much more than a camera. It is a kind of a camera with a very good and powerful image processing. So the retina is very complex structure. It is actually part of the brain. It's a kind of an extension of the brain going out of the skull. And it's the most important sensing organ. The eye is the most important sensing organ in the body. Like about 90% of the information that we gain from the world is from vision. And actually, if you think about it, it's very interesting because many expressions in language are dealing with vision. For example, in Hebrew, mi hu hachacham haro'eh et hanolad. Yeah? Right. And in English, if I ask you something, you say, yes, I see. It is very clear, or it's vague. I don't see it. Yeah? So we use the terms of vision to say that we understand and we are clever and all these kind of things. So vision is very important. So in the retina, which is in the back of the eye, it's the beginning of the vision process. So there are about 100 types of neurons, cells type in the retina. Each one has different structure and different function. So in the very back of the eye, there are the photoreceptors. So the photoreceptors sense the light, and there are detectors, and they are very highly specialized detectors. They can sense, some of them can sense 1 photon of light, much more than any camera that is available today.
Wait, these photoreceptors are part of the retina?
Yeah, they are in the very back of the retina. So they are the first layer that the light comes and being detected by the photoreceptors. There are about 120 million photoreceptors. Most of them are called rods, and they are being used for low lighting conditions. So they are mainly in the periphery of the vision of the retina, and they help us not to bump into structures, and they are very important for night vision. There are about 95% of the photoreceptors. There are about 5 or 6 million photoreceptors, which are called cones, and they are very important for color vision, for fine structure detection, for reading, for face recognition, and they are in the center of our vision. So we have like 2 kinds of vision. In the center of our vision, we can detect faces, read, and see very fine structure. And in the periphery, it is useful not to bump into things when we are walking, and to know when a car is coming, and to look at, to change our gaze toward it.
So when people have issues with vision, it could be in either space.
Yeah, exactly. So some diseases like AMD, age-related macular degeneration, which is the leading cause of blindness in the Western world is affecting only the central vision. So people cannot read, cannot recognize faces, but you don't see them, walking with a
They're not
dog.
bumping into things.
Yeah, exactly. And the peripheral vision, when there are some diseases like retinitis pigmentosa, they are in the periphery of the vision. People can, at least at the beginning, read, but they bumped into things, they cannot drive, and eventually, in some cases, they will lose also the central vision.
So your laboratory that's creating and developing this extraordinary miniature device for the retina, what is it addressing? Which of these issues? And how does it work? What does that mean to implant a miniature device in your eye?
So actually, since the central vision is of high acuity and the peripheral vision is of low acuity, it's a different approach toward a restoration of sight of these two kinds. And I could say that it is much more challenging to restore the central vision because you want to be very close to natural vision. So in natural vision, the resolution of what we call 6.6 or 20/20 is the resolution over about 5 micrometers on the retina. So it's a single cell resolution. So we are at the moment trying to solve this problem of very high acuity of the central vision and not the peripheral vision. So our device and our work is aimed at restoration of sight, of the central sight of AMD patient or some other diseases that are losing the central vision.
How does it work? Again, that all sounds incredible because as you said, you started with losing your vision is traumatizing. It's a real handicap for somebody. But what does that mean to implant a miniature device? What size? What are we talking about? Do you feel it in your eye? And then how does it work scientifically? What is it you're really doing with it?
So we have to go back to your first question because you wanted to know a little bit about the eye structure. So generally speaking, if we want to restore vision, I think that our philosophy is to mimic the natural vision as much as possible because this is amazing and complex machine. As I said, it's a kind of a camera together with a highly powerful image processor. So if we go back to the structure of the retina, so there are the photoreceptors, and the photoreceptors, they are sensing the light, and then they transfer the information to the next level of cells, which are called bipolar cells. They are called bipolar cells. And they do image processing, a very powerful image processing. And then the information will go to another cell, which is called the ganglion cells. And the ganglion cells with some other types of cells will do another complex image processing. And then the ganglion cells will digitize the information. It will make it like a digital mode of operation, like what we are doing now. We are recording this conversation and everything is becoming into numbers. So the ganglion cell will actually code the visual information into a kind of a code which is digital code. It's a kind of 0 and 1.
Really?
Yeah. So the ganglion cells operate in a kind of mode which is called action potential. Action potential is a 0 or 1 event. It's a non or action potential. And this is actually, most of our neurons in our brain operate in a kind of this digital information. They operate in a 0-1
like
mode.
a computer.
Exactly like a computer. And actually this is what made, people build all this artificial vision today, and artificial intelligence and AI and everything that people are doing today is actually inspired by our brain.
That processes in when you say 0-1, you mean it either is or it isn't?
Yes, exactly.
Okay.
So the ganglion cells in the retina, the very last cell in the retina, digitize the information and send it in a kind of a digitized way to the inner part in the brain, to the visual cortex. Our visual cortex further do complex processing of the information. Now, if you go back to the retina, the very first two cells, the photoreceptors and the bipolar cells, do not operate in a digitized way. they work like an analog amplifiers, analog processors. And if people are, some people who are very interested in music, they always want to have this analog amplifier. Back in the 60s or 70s, people had very large amplifier because an analog amplifier can code tiny amount of information and changes like in your voice and in music and also in vision, even better than digital information. So the very first operation of the retina is an analog mode of action, and then it is being digitized.
So does that mean it's like a little more nuanced because it's not 0-1?
Yes, exactly. So, if you want, so our vision is so delicate, so we can see, tiny change in illumination. For example, if you look at the expression of someone when we are talking to someone, you see tiny change in his, in his lips or in his skin because of less than a percent, 0.5% of even less of amount of light is coming from a specific structure. And then you see that he is smiling or is not smiling. And you can detect contrast of 0.5%. It is amazing, more than most of the cameras today. So in order to do this, you have to be in an analog mode of action in vision. And then the information is being digitized. So another thing is that the retina has a lot of circuits. It is like a computer, as I said. And for example, we have some circuits to analyze color. So some circuits will try to compare between the red and the green. And some will take and try to compare between the blue and the green.
So this is where color blindness could come in?
A little bit, yes.
Or am I throwing you off? Let's stick with where we are. I've got a million questions, but we'll stay focused. It's fascinating.
For example, some cells in the retina will respond to increase in light, and some will respond to decrease in light. So actually, when we are looking at something, our eye is doing what we call active sensing. It is not like capturing an imaging. It actually, because we have tiny eye movement, it is like we are trying to sense. Like if you try to find anything on your ear and you try to see, you will not touch it like a static. You will do something like this. So this is exactly what the eye is doing. It is like sensing it by slowly, very fast and tiny eye movements. So it's an active sensing. So if we want to restore vision, we need to know everything about the eye and the function and try to mimic it. So how does it work? So in some of the blinding disease, in some of the retinal degenerative disease, there is loss of only the photoreceptor. Only the photoreceptors are being lost, but the rest of the retina, all other structure, the bipolar cells, the ganglion cells, some other cell, amacrine cells, there are so many other cells, they are still preserved, at least relatively preserved. And the cornea, the other structure of the eye, and the optic nerve are all preserved. So what we are missing is only the photoreceptors, which are the sensing parts.
And so you say only, but it's huge. You started out explaining that it's significant.
It's significant, but we still, if we have a way to stimulate the bipolar cell or the ganglion cell and bypass the problem, then we can restore vision. And some people want to try to do it by directly stimulating the visual cortex in the brain, and then you don't need an eye. Right. I mean, and this is what people are now trying to do. But if we talk about what would be the best way to stimulate this visual system is to stimulate it at the earlier possible station. So the bipolar cells can still preserve some of the processing of the image. So it's better to go from the bipolar cells, and then the information will be transferred to the ganglion cells and to the visual cortex. So how can you stimulate a neuron? So we all know that if we accidentally touch electrical power, then all our muscles are being twitched. So it is not recommended to do this, but we can send electrical pulses to the bipolar cells or the ganglion cells and stimulate them by electrical power, electrical field. There are some other way to do it, but we don't have now time to do it. There are some things like optogenetics and some other things that can stimulate the remaining neuron of the retina. So what we call a retinal prosthesis is actually inserting an electrode array, an array of electrodes below the retina or above the retina to stimulate the bipolar cells or the ganglion cells. And we prefer stimulating the bipolar cells.
Because they're earlier on in the process, as you said.
Exactly, so we can preserve some of the processing. And then if you have a kind of an array of electrodes, let's say 1,000 electrode, 10,000 electrode, and you can stimulate them in a kind of a patterned manner. Like if you want the patient to feel a line, then you stimulate a line of electrode, and the patient will tell you, I see a kind of a line. If you will do a one electrode, the patient will say, I see a tiny light. If he feels it like a kind of a light, it's not colored, because we are actually losing the color vision because we don't have these photoreceptors, and it's very challenging to restore the color. But the patient can get something which is a kind of a black-white or grayish mode of vision.
Which is a lot for somebody who is currently unable to see anything.
It's a lot. And some of the patients can benefit, and there are hundreds of patients already implanted with devices that were available in the past, and they were not so successful, but they paved the way toward better vision. And this is what we are now trying to do, is to take what is currently available and to go to the next level of visual restoration.
And these are at a nano size. They're tiny, tiny, tiny.
They are very tiny. They are in the micro size, and we are trying to overcome the current limitation of retinal prosthesis. So there are at least 3 main limitations. And I would say that the limitations are because the current retinal prosthesis, they have inherent limitation which does not enable them to mimic the natural vision. So the first is that, in order to get a visual acuity of 6.6, or like our natural 20/20, we need to stimulate 5 micrometer on the retina. And this is like a single cell. Now, what is happening is that even if you have an electrode which is 5 micrometer size, because of the distance between the electrode and the neurons that we want to stimulate, the bipolar cells in this case, the electrical field is spreading. So it's not like a laser. A laser will go like this. Electrical field will go to the sides, and then instead of stimulating one cell, we will stimulate like thousands. So eventually, even if the electrodes are 5 micrometer size, and we can do it, this is doable, the actual resolution on the retina will be very low. So currently, the resolution on the retina is at least 25 or 100 times less than what we would like to have at the moment with current retinal prosthesis. This is one thing because of the spreading of the electrical field. Another thing is that we discussed this, the complex circuitry of the retina and the processing. So with electrical activation of neurons, we activate all the neurons at the same time. So we are losing the color, we are losing the differentiation between this, so to speak, on cell and off cells, which, some of them are increasing their activity, some of them are reducing their activity. So we are actually doing some kind of, non-selective activation of the retina with the electrical field. And lastly, the third limitation is that we discussed this analog mode of action. So with electrical field, you cannot have this analog mode of action because of some technical constraints. So we are forcing the cells to behave like in a kind of a digital mode of activation. The bipolar cells.
The ones that should be analog, you're doing them more digital.
So we are losing this fine structure. So for example, people with retinal prosthesis, they don't have like 200 grayish level, like what we have. 200 level of grayish. They have about 4. So it's almost like yes or no, black and white. So they can see that maybe there is a glass here, but they will not see the fine contour of the fine structure.
It's like the way I would see it without my contact lenses.
Exactly, yeah.
It's not fun. It's not fun, but if I couldn't see anything, I'd be thrilled with that. Yeah. So how close are you to, let's start with addressing some of these problems. This is where your research is going. If you were to project into the future, and I know you can't really do that as a researcher, you're always hoping every day is going to be a eureka, but realistically, how far away are you from really getting this to, even if it's not 100%, getting it to a significantly improved state?
So what we are trying to do is very complex, because actually, you know, I always say that engineering and physics are, you know, very complex to do, to understand, but at the end it works. But biology is very complicated. And what we are trying to do is to combine engineering and physics with biology. So, you know, a simple, I mean, this is not very simple, a simple retinal or conventional retinal prosthesis is stimulating the cells with electrical field, and it works. It also works, but it's still not what we would like to have. So we are complicating this and adding some kind of biological structure to this. We call this hybrid retinal implant. So what we are actually doing, we are fabricating an implant that is composed of tiny electrodes, which are 10 micrometer size. The 10 micrometer size, and they are situated in a microwell. The tiny microwell, which is a 10 micrometer diameter. So 10 micrometer diameter, it means that it is about tenths of the width of a single hair.
Wow.
Yeah.
But you said the little cells are 5 micrometers, right?
The cells are, the electrode are 5 micrometers. The microwell is
10 centimeters.
10 micrometers. And in each one of these microwell, we insert a cell, biological cell.
That you've produced in a laboratory?
That we produce from human embryonic stem cells. And the cells are actually serves as a kind of a translator or mediator between the electrical field, which is not the language of the retina, and they translate it into the language of the retina, which is, we call it neurotransmitter, like neurons. Neurons operate one between other with a kind of a chemical language, which is called neurotransmitter. So our cells, which are part of this hybrid implant, are actually translating the information into what the cells are used to work. And by this, we can first preserve the retinal circuitry, these things of the on, off, and maybe some other circuitry. Also, because of this fine structure, we are actually confining the electrical field to a specific area and preventing the spread of it. And by this, we can have very high visual acuity, at least according to our calculation. We have shown some of this. And also, by this structure, we can get into this analog mode of action.
I was just going to ask you if it addresses the issue, because you have now the cellular structure that would enable it to potentially translate it back into the analog and not just keep it, you know, the electrical. Your cell translates the electrical, but it's giving digital back into the analog, which will fix a lot of the issues, it sounds like.
So we are trying to overcome the current inherent limitation and trying to mimic the vision, the natural vision, as much as possible.
How far along are you with this?
So this is a very long run. So I can tell you that it took about 20 years to get into the point where patients were implanted with retinal prosthetics with the conventional one. We are working for this project for a few years, more than 5, and it will take some more before we can get into human. So we are currently at animal work, preclinical work, and we have very good results. And yes, we are now thinking what would be the next stage. We have some challenges like transferring the amount of energy that is needed into these tiny cells. It is very, very complex because you don't want to have so many wires getting into your eyes. So this has to be with some kind of wireless way to do this.
Very complicated. So Is it the same for every patient, or is this where you're associated with the Dangoor Center for Personalized Medicine? Does each device have to be personally attuned to each individual as you move forward? Obviously, you said you're not yet in human trials, but when you get there, even on the animals, can you know, obviously you're not going to mass develop anything, but can you create a number of these and stick same one in everyone's eyes? Where does the personalization come in?
So generally speaking, if we are thinking about diseases that affect the eyes, there are so many. So you have to find what would be the best for each patient. So if we go to, so AMD is age-related macular degeneration. This is the most common thing. And there are genetic disease in more than 270 genes.
Wow.
Yeah, each patient will have another disease. And for each one of these diseases, there are other solutions, but there are some common solutions. If we will talk a little bit about gene therapy, you know that there are some people, very excellent scientists in Bar-Ilan that are doing. Each gene, each patient has its own mutation. It needs a different correction.
Yes.
And it's the same with retinal diseases. And we are trying to work also with some delivery of this kind of things into the eye. The beauty with retinal prosthetics is that it can solve the problem of many types of genetic disease. So it's a one solution to all, but with our cells that are being on board, it would be the best to have the patient own cells on board.
So there's no rejection issues.
There is no rejection. So today there are some technologies that enables you to take the patient's cells, for example, from a skin biopsy, to take the fibroblast from the skin, grow them in lab, and then make them into the patient's own photoreceptor precursor or something like this.
Wow.
Yeah.
So do you have colleagues with whom you work, I assume, at Bar-Ilan, other universities? This is such a complicated process. Is this something, as you're working on this device, are you working across Bar-Ilan? Are you working outside of Bar-Ilan with other researchers, other PIs, and internationally?
Yeah. Most of the work that is being done with this retinal prosthesis is done in my lab. We have a collaboration with Professor Zalewski from the engineering.
Bar-Ilan's engineering.
Yeah, and everything is at the moment being fabricated at the Nano Center of Bar-Ilan University.
Wow.
Yeah.
So it's all internal.
Yeah, it's internal. We have some collaboration with scientists in Stanford and in Berkeley, but not on this specific project. But there are some projects which are, complementary.
How did you get to this? I'm just going to back up a minute. This has all been amazing, but I know you're trained as an ophthalmologist. You're a physician.
Yeah.
How did you veer into research?
Yeah.
Why aren't you just, doing surgery on people's eyes?
Yeah.
How did you land where you are?
Yeah. So actually, when I was young, I thought that I will be a physicist. I like physics and to know how things work, but I wasn't sure that this is what I'm going to do. And eventually, I was drafted to the academic reserve in the military as I studied medicine before the military. I wasn't sure what I'm going to do, but my father was a physician, so I was exposed to this, and then it was very interesting, and I was in the military for many years, like in the battalion and in division, and then in the last few years, I was heading the research and the foreign affairs directorate of the IDF medical course and the Ministry of Defense, MAFAT, yeah. MAFAT, it's the Directorate for Research and Development. So I was exposed to many things with research, aimed at helping soldiers and everything that has to do with security and medicine. And then I was loved with this, with the creativity in research. And then I found that I could combine my love to physics and engineering and all these things with medicine and ophthalmology. So I said, okay, I have to do a PhD in bioengineering. And I was very happy with this. And I think that today, what I'm doing today is everything that I've ever learned is with this project. Like,
it does. It seems like it's pulling from everywhere.
Everywhere.
And why did you choose Bar-Ilan? I mean, now that you're telling me a little bit about it, sounds like you have professors, you have your lab, you're the head of the School of Optometry and Vision Science, you're associated with the Nano Center, but look, you studied at Stanford. No. You have incredible credentials. What made you choose Bar-Ilan and what made you stay at Bar-Ilan University?
No. So yeah, I had a few other opportunities, but I chose Bar-Ilan mainly because I like the people, the atmosphere. That's the most important thing. And you can feel it from the beginning. And also because we had the opportunity to build this School of Optometry and Vision Science. So I always wanted to build something, not just to join a thing that an established faculty. So me and together with Professor Uri Polat, we were like the founder of this school, and we worked many years together. He was the head of the school, and since last year I'm the head. So I found it very interesting and challenging to build this school. So today we have like a bachelor in science in optometry, and we have master in optometry, and master in optometry, clinical optometry, and master with thesis in vision science and graduate studies, PhD in vision science. We have like about 50 scientists and students and postdocs.
That's a lot.
Yeah, that are working in the field of vision. So I think it's the only center in Israel that has such a focus on vision, and I like it very much. So it's a good excuse to be in Bar-Ilan University.
But it also sounds like you have colleagues with whom you work, and Bar-Ilan does offer, as you said, the Nano Center, and you have Zev, who's amazing in engineering, who also focuses on some of the vision things.
And we are part of the Faculty of Life Science, which is a great, many collaborators from there, and it's a good house for us, a good home.
Where do you see this going? Meaning you're going to work on this. You said it's going to take a little bit of time to get this to human trial, and then hopefully it'll go to market, and you will help millions of people around the world. What do you expect, though, in the field of vision science in the next 10 years, say?
So I think this field of vision science and vision restoration is like booming. It's an exciting period now. I think that, the eye was always of high focus for research because it is so important, vision, and it is very accessible. For example, the first gene therapy that was approved by the FDA was for the eye problem, for a specific mutation in the retina, another cell, the retinal pigment epithelium, near the retina. And currently there are dozens of gene therapy trials. So the eye is always at the leading edge of the science. So it's exciting to see how many things are now happening. So I think that in the coming years, we will see more and more drugs that are being, or gene therapy approaches or strategies are being approved for retinal diseases. This is one thing. With retinal prosthetics, we have the advantage that some of them can fit all. I mean, because for gene therapy, you need a specific gene, so it takes years to approve it. With retinal prosthetics, one of the advantages that it can fit all, but with our device, we will need to do some more modifications.
Right. So it's personalize a more generic device.
Exactly. So you need to balance, because if you are highly personalized, then you are losing some of the patient.
Right.
So it's exciting time. There is also an approach of optogenetics that people are trying to do. And there are some other things like all eye transplantation that we are now going to be part of this.
Really?
Yeah.
That's after post-mortem, someone donates their eyes?
Exactly. Exactly. So this can fit. And there are some people who are working on transplantation electrodes in the visual cortex. So in this case, even patient who doesn't have an eye or is damaged eye, for example, people with trauma, after trauma to the eyes, then retinal prosthetics cannot help them. But if you stimulate the visual cortex, then you can restore vision. However, currently the results are not so great because it's only the beginning of this project. But maybe in a few years it will be better.
It's amazing to look forward to see where we're going, and that you're part of this. And I wear contact lenses, so the idea of implanting something in my eye is both, I'm aware of how challenging it can be, and I'm aware that it's quite possible. And the miracles that have taken, they're not miracles, obviously they're scientific discoveries, but for those of us whose vision has been restored, it feels miraculous. So thank you for joining us today for this conversation. You opened my eyes, if I could be a little punny here, to the whole world of vision science and the amazing work that you're doing in your laboratory that will help so many people into the future.
Thank you, Sharon. Thank you.
Thank you.